Next slide is about the packaging process. Before going to discuss about the packaging process, we know about the mixing. And the sterilized carrier material is spread in clean, dry, and sterile metallic or a plastic tray for 2 to 3 days at room temperature. Before packaging, this process may happen, that is called a curing process. Curing, which improves the cell count up to the level of 10 power 9 to 10 power 10 cells per gram. So curing is a process just spreading the carrier material on the metallic or the plastic tray to improve the cell count. That's why we follow this curing process. Then the bacterial culture is added to the sterilized carrier and mixed well by manually or by mechanical mixture. The culture suspension is to be added to the level of 40 to 50% water holding capacity. This is called a moisturizing capacity of the carrier material or the moisture content of the carrier material. So the inoculants are now packed in a 200 gram quantity paper of polythene and sealed with electric sealer and allow for curing at room temperature. One more time the curing process may happen, but here we are sealing the packets and then the curing process may process. Next slide is about the specification of polythene bags. Some of the specifications may be indicated as well as the regulations are followed before packing this carrier material into the plastics. So the polythene bag should be of a low density grade. This is the first rule. The polythene bag should be of the low density grade and the thickness of the bag should be around 50 to 75 micron in size. Next, the polythene bag has commonly used as a packaging material following impregnation of carriers. So the polythene permits high gas exchange as well as allowing for CO2 losses and O2 uptake. Sometimes the polythene bag may releases some CO2 as well as uptake some CO2 from the air. Thus, over the 6 month of period of storage, almost at the 26 degrees Celsius, it was found that the moisture content of the ash declined from 50 to 45 percentage. So, each packet should be marked with the name of the manufacturer, name of the product, strain number, and the crops to which the recommended method of inoculation and the date of manufacturing, batch number, date of expiry, price, full address of the manufacturer, as well as the storage instruction. These are also included in the specification of the polythene bags. Next slide is about the storage of biofertilizer. Here the packets should be stored in a cool place away from a heat or direct sunlight. The manufacturer may store the product at 4 degrees Celsius for any period up to the expiry, but is restricted to the maximum of the period of 6 months after release from the store. So the population of inoculant in a carrier, the inoculant packets may be determined at 15 days of interval. So the population growth density, it may be the interval period for the growth doubling time is 15 days. There should be more than 10 power 9 cells per gram of inoculant. At the time of preparation, we have a 10 power 7 cells, but after the first period of interval, it should reach the 10 power 9 cells per gram. During this transportation, you must keep the organism alive. Right combination of carriers should be used. The other chemicals should not be mixed with these biofertilizer packets. So the shelf life of the carrier, it's almost based on the transportations as well as the storage also. It's about 6 months to 2 years for the liquid formulation. The 6 months is efficient for the granular material, but the 2 years period is maybe for liquid formulation. This is the storage method of biofertilizers. Next slide is about the biofertilizers effect on a soil fertility. Here, the autochthonous soil bacteria, which means indigenous or the native bacteria, enables the soils to cycle the nutrients and water to maintain the healthy structure of the soil. So, they also promote the plant growth. Even under a stressful condition, the bacteria can able to promote the plant growth. Sometimes the soil lacks a life-giving nutrients, almost organic matter or NPKs, nutrient of nitrogen, potassium, and then phosphorus, as well as the rich microbial communities. Sometimes the soils may lack these are all the factors. At that time, the microorganisms need to regular supply of active SOM. SOM means soil organic matters. Okay, SOM means soil organic matter, which provides the nutrients, organic matter, rich minerals to the microbial community. So the SOM is a organic substance as well as anything about the carbons. These are called SOM. So the carbon in a soil, both living as well as the dead plants, provide the carbon to the soil structure. They also increase the soil fertility by incorporating the air, minerals, and the nitrogenous compounds. They contribute in increasing the plant growth by providing essential elements, minerals that plants cannot utilize by their own. So the microorganism decompose the organic matter into simpler form that can be easily uptaken by the plants. So the biofertilizer is a useful product for economically weak farmers who are unable to promote the efficiency of the nitrogenous and the phosphorus sources in the soil. So the formulation of the quality check and the application methods and the soil managements are essential for biofertilizer quality maintenance. Next slide is about the factors affecting the efficiency of biofertilizers. And the factors are called soil temperature, biofertilizer sources, species, soil aeration, carbon nitrogen ratio, ventilation, soil moisture content, and the degree of reaction of pH of the soil. These are all the factors affecting the efficiency of biofertilizers. Next term is about NUE. NUE means nutrient use efficiency. It's a critically important concept in evaluation of a crop production system. The evaluation of NUE is useful to differentiate the plant species, genotypes, and cultivars for their ability to absorb and utilize the nutrients for maximum yields. It can be greatly impacted by the fertilizers management as well as by the soil plant water management. So the objective of the nutrient use is to increase the overall performance of cropping systems by providing economically optimum nourishment to the crop while minimizing the nutrient losses from the field. So the nutrient index, next term is about nutrient index. Here the NI value is a measure of nutrient supplying capacity to the soil of to the plants. This index is used to evaluate the fertility status of soil based on the samples in each of three classes, particularly in low, medium, high fertility areas. So the NUE means nutrient use efficiency method. Second index is called nutrient index method. These two methods almost measure the capacity of the soil as well as the capacity of nutrient uptake by the plants also. Next slide is about the quality checking method for particularly the Pseudomonas as well as the Azoospermum. In a Pseudomonas, the quality control almost begin with the maintenance of mother culture. And the broth culture, almost the mother culture is in a liquid medium, so it is called broth culture. This broth culture is added to the carrier material. Before adding to the carrier material, you must check the count of the organisms. The number up to 10 power 7 to 10 power 9 cells per gram, it's sufficient to mix with carrier. And next one, Azoospermum. Here the Azoospermum quality control test, it may be subjected to packaging, the time as well as the storage. So the Azoospermum quality, it must be checked at the period of storage as well as in the packaging timing. So the Pseudomonas checking method is by adding before to the carrier, and the Azoospermum, it must check the quality before packaging as well as the storage of organisms. organisms. So the BIS means Bureau of Indian Standards. Here the BIS norms are also called as standards for the biofertilizer or the standards for the existence of biofertilizers. Here the carrier must be in a moist condition or a dry powder, or sometimes it may be in a liquid form. The pH of the carrier is about 6.5 to 7.5. It's a constant one. Next particle size is about 0.15 to 0.212 mm is a natural range of the particle size. Moisture content, it must be in the range of 30 to 40 percentage. These are all the factors almost we must follow before the packaging of carrier into the packets. Next slide is about the checking method or the quality checking method of biofertilizer. The first method is called a plate count method. There are so many methods followed in a quality checking. First one is about the plate count method. Here the capital cost for this plate count is a low amount, and the variable cost, it may be also low, and the time to developing this plate count is very fast compared to other method, and the assay timing is very slow, and the specificity means nothing. The carrier bind with the or inoculant, it's called a specificity. So the combination of a carrier with the organism, it's called specific nature. It's a low method, low one. And the final one is a measure of viability. Viability means the cells may in live condition or in a dead condition. Here the cells are in live mode. That's why the plate count is efficient one. Second one is a selective plate count method. Here the capital cost is almost low and the variable cost is also low. Time to developing this process is almost fast, similar to the plate count. In assay timing is very slow and the specificity is medium compared to the plate count. And the viability is also live in nature. That's why the selective plate count is also an efficient one. The plate count, the first one plate count is based on the nutrient agar method, but the selective plate count is based on the particular method. For example, the pseudomonas prefers asphys medium for their growth, and the azospirillum requires some other minerals for their growth. That's why it's a selective plate count method. And the multiple tube fermentation method is called the MPN test. Here the MPN test capital cost is low. Variable cost is also low. Time development for this method is almost fast. Assay timing is very slow, and the specificity is about the medium, and the viability measurement is almost live in conditions. So these three steps are called enumeration processes. Next one is advanced method. It's called indirect ELISA technique. Here the capital cost is medium, viable cost is low, time to development for this method is slow, and the assay timing is very fast. Specificity is also high, and the measure of viability, it includes dead cells. In a ELISA technique, we can count the number of viable as well as the dead cells. There is no differentiation for dead cells from dead cells identification from the live. So it is a combination of dead cells as well as the live cells. Both cells are present in the ELISA method. It's almost in an indirect ELISA method. In an immunoblotting technique, the capital cost is medium, viable cost is medium, time to developing is very slow, assay method is very slow, and the specificity is very high. ELISA is also higher affinity, and the immunoblotting technique have a specificity is high compared to the remaining techniques. And the measure of viability is live cultures only included in this technique. In a sandwich ELISA, we have a capital cost is medium, variable count is low, time to developing is very slow, assay timing is very fast, but specificity is also high. But here we also have a live as well as the dead cells, like indirect ELISA. Last one is about the quantitative real-time PCR. The capital cost for this method is very high compared to compared to the other methods. The variable cost is also high. Time for the developing it's very medium, and assay timing is very fast. Specificity is very high, and the factors affecting the accuracy is almost the sensitivity calibrations, and the viability is also Dead cells DNAs may incorporate the results. These are all the checking process of biofertilizers quality. And after quality checking, we must to follow the efficacy. So next slide we can discuss about the biofertilizer efficacy in details.